question. There's two questions here. One, which parts of the brain were you looking in and why? And then question two is, how much DMT did you find there?
Right. So we were interested in the prefrontal cortex and the somatosensory cortex. And prefrontal cortex, I think, for obvious reasons, because of its role in executive functioning and high expression of serotonin 2A receptors. And the somatosensory cortex was a region that our group had been interested in. It was a region that our group had been interested in for the past several years. We had done different experiments with ketamine and nitrous oxide, and we saw some interesting trends with the somatosensory cortex in terms of acetylcholine levels. So those were the two regions we were interested in, and we were measuring them not with a traditional microdialysis probe, but with a open flow microperfusion probe. And this is kind of an important part of the study because it allowed us to actually improve the analyte recovery and get kind of a better picture of what's going on neurochemically in those regions. And we had trouble in the past measuring the levels of DMT in these brain regions. But we thought that if we're administering DMT intravenously, then that's going to cause a massive spike in the brain. So this is going to allow us to sort of develop a method to be able to detect DMT in the brain, because it's going to be such high levels, it'll be very easy to detect. So after we started administering DMT IV, we saw huge spikes in DMT, and we're like, okay, now we know what the DMT peak looks like on our HPLC. So we were like, okay, let's try and measure it during baseline conditions now, because now we've got the method worked out.
I see. So flood the brain with a lot of DMT so it's easy to detect. You can fine tune your method for knowing how to see it when it's there. So now you're going back to baseline conditions and now you've got a really good tool so that you know it when you see it, if it's there.
Exactly, yeah. And that was some of the trouble in the early days when we had the INMT knockout rats, because the first experiment is like, well, why don't you take the knockout rats and measure the DMT levels in their brain? Well, we couldn't do it. It was so low, we just couldn't quite get that measurement worked out. But later on, when we started administering the DMT IV, yeah, like you said, we had a clear method, and it was much easier to do. So at that point, we had this open flow microperfusion, which is another benefit of the study, allows us to kind of improve recovery. And with that, we were able to measure DMT under baseline conditions, and then we were able to track it throughout the entirety of the experiment after we administered it intravenously.
And then I'm doing a screen share here, so we're going to be really careful to describe this verbally because most people are just listening. But what are we looking at here? How much DMT did you actually find in the prefrontal and somatosensory cortex? How did that compare to other things like serotonin and dopamine?
Yeah, so it pretty straightforward. The levels of DMT fell right within the range of serotonin and dopamine in both brain regions. So with the plot that we're looking at, this is basal concentrations of DMT in nanomolar levels. And it's showing serotonin, dopamine, and DMT for both brain regions, prefrontal and somatosensory cortex. And with serotonin, the basal levels looks like right around 0.8 nanomolar. With dopamine, right around 0.4 nanomolar, and then DMT fell right within that range, about 0.6, 0.7 nanomolar in the prefrontal cortex. And then pretty much the same trend in the somatosensory cortex too.
So this measurement, so what this is really showing us is that, and is this rats or mice? I forget.
Rats.
So in the rat brain, in prefrontal cortex and in somatosensory cortex, you can detect DMT at levels that's comparable to two other major neurotransmitters in the brain.
Right.
And this is basal conditions. So the animals are alive, they're awake, they're not doing anything in particular. You're not giving them DMT, and there's sort of nothing special happening, just baseline conditions.
Yeah, exactly. Yeah. We just keep them awake. Like you said, yeah, they're alive. We just don't allow them to sleep. And yeah, just the baseline conditions. So this is actually, I think it's one of the bigger findings of this work. But it's basically just supporting what was shown in the previous paper. So it's already been shown by John Dean's work that DMT is present in the brain at about those levels. One of the differences is that John Dean and the others, they were measuring an occipital cortex. This is in both frontal and sensory cortex, which is the first measurement in those brain regions.
Yeah, and I mean this would imply that, like plausibly, it's just found throughout cortex.
Right. Yeah. And this would support the work from John Dean showing that INMT is expressed widely throughout the cortex too.
Mhm. Okay, so the next figure, I'll put this up on the screen as well. This is going to be an experiment where you're actually now giving animals IV DMT, and you're looking at DMT levels across time. So it looks like you use three different doses, and I'm hoping you can just unpack this result for people.
Yeah, so we're giving three different doses here, and the low dose was 0.75 mg per kg, milligrams per kilogram. Medium dose was 3.75 milligrams per kilogram. High dose was 7.5. And what we did is, I mentioned previously that we were measuring the DMT in these 12.5-minute epochs. So it takes 12.5 minutes to collect one sample. And the first four samples are baseline, so just basal conditions. And then the next seven samples are drug conditions. So the DMT is administered at the beginning of the fifth sample, and it's just a 5-minute bolus infusion intravenously into the jugular vein. And we measure the DMT in that first sample there, and then all of the additional samples following that.
I see.
And yeah,
So each of these data points is about 12 and a half minutes of time.
12.5 minutes, but it's there's a number of different rats for each data point, obviously, too.
Yep.
So
Yep. Okay. All averaged together. And we're looking at, in this case, we're looking at DMT levels at each point in time, each 12.5 minute chunk, average across multiple animals. We've got three different doses represented here. Talk to us about what's happening here to DMT levels. And then can you explain that in relation to the behavioral effects and how long those last?
right. So unsurprisingly, we see this dose-dependent increase in DMT levels in the brain because we're administering it into the vein, and then we're measuring it in the brain. You think the more DMT you give, the more you're going to be able to measure in the brain. And there's a large and significant spike at all three doses during the drug infusion period. Following that, we sort of group the epochs into drug, post-drug, and recovery. So that just to kind of make it a little bit easier. The post-drug encompasses the D3 through D5 epochs, and then the recovery is just the D6 and D7 epochs. And there's also sort of a dose-dependent sustaining of the levels of DMT in the brain, where at the low dose during the final three epochs and in the recovery period, we see that the DMT levels go back to basal conditions, so they're no different from the baseline conditions. But in the medium and high doses, those levels of DMT remain significantly elevated from the basal conditions. And this is true in both the prefrontal and the somatosensory cortex. And this is interesting because if we say each epoch is about 12.5 minutes, and the DMT was administered here at D1, or at the first drug epoch, there's 7 epochs there times 12.5 minutes. That's maybe about an hour and a half. And that indicates that when DMT is administered intravenously, 90 minutes later or more, we can still detect high, significantly elevated levels of DMT in the brain. And this is interesting, I think, because this is not the trend you would see if you were measuring DMT in the blood. If you administered DMT, like this is the way that it's done in the human studies. You administer DMT into the vein, and then you're also measuring the DMT from circulation as well. And you see that DMT is cleared from the blood much faster than it is from what we see here in the brain. And that suggests that there could be some potential storage or sequestration mechanism in the brain that's allowing the DMT to remain significantly concentrated there for a prolonged period of time.
I see. So it doesn't prove this, but it suggests the possibility that there's some way to store DMT in the brain for extended periods of time.
Right. Yeah, because this is not what you would expect if you were measuring DMT in the plasma or in the blood.
Yeah, it would spike and then drop off much more quickly, probably within these first 2 time points, right?
Right, exactly.
And Okay, interesting. How long? So, if you're just assessing the animals behaviorally, so let's say you're looking at the classic head twitch response in animals, which is our proxy for hallucinogenic effects. What would that look like in comparison to this? So for example, at D3, D4, D5, especially at the higher doses, you've still got elevated DMT levels in the brain of these rats. Do they look behaviorally normal at that point, or do they still have a head twitch response or other indications that they are tripping, basically?
Right. So yeah, thanks for bringing that back up. The behavioral response was also dose dependent, and I think what I wrote in the results was that with the low dose, the behavioral effects last approximately 10 to 15 minutes. Medium dose, maybe 15 to 25 minutes, and then the high dose, up to 35 or 40 minutes. And the head twitch response is actually a very, very acute response. It happens in the
Mm,
first 10 minutes from the start of DMT administration to 10 minutes after that. After that, you won't see any head twitches. So they all occur in that D1 period there.
I see. So the classic head twitch response that people use to assess something's hallucinogenic potential, that would all be occurring in this first 12 and a half minute D1 time period here. Do the animals do any other behaviors that indicate that they're having a drug effect after that time point?
Yeah, so those are the dose dependent effects that I just described, the different durations there.
But
Um,
what exactly are they? How do you tell? What are the behaviors?
right. Yeah. So the animals will kind of we call it pancaking. They'll kind of splay out. Their hind legs will be splayed back. There will be some head weaving, so the head will kind of rhythmically weave left and right. There's they call it forepaw treading, so they're kind of like digging with their front paws. There's sometimes this phenomenon of like straw tail, where the tail kind of curls up, points upward, becomes erect, and almost points forward. And then sometimes even some backward walking at the very high doses.
Hmm.
And some of these behavioral responses have been noted in serotonin syndrome or excessive serotonin receptor activation. So it could be related to the
Mm,
excessive high dose causing overactivation of serotonin receptors.
I see. And maybe we'll come back to this later if we have time, but I'm just going to say this to put it in my own memory. What you're saying is there are clear behavioral indications of the hallucinogenic effects of a drug like DMT, even when the animals aren't engaged in a head twitch response.
Well, you said hallucinogenic effects, and we don't know that the animals are hallucinating. So there's behavioral effects,
Mm.
but we're not sure if they're hallucinogenic.
Yeah, let me say that. Let me say that a slightly different way. The head twitch response is presumed to be a proxy for hallucinations that would occur in humans when we study these things in animals, who we can't get inside the minds of. But there are reliable behaviors these animals will display to known human hallucinogens, um, even in the absence of head twitch responses.
Right. Yeah.
Okay. Okay.
Yep.
Interesting. Okay, maybe we'll come back to that. But okay, what is this next experiment showing us here?
Next one here is looking at the effects of IV DMT on the levels of serotonin and dopamine in both of these brain regions.
I see. So you're basically asking, if you give an animal DMT, does it change the levels of other neurotransmitters in the brain?
Exactly. Yeah. And with these two neurotransmitters specifically, it does, particularly for the medium and the high doses during the drug effect. So it's an acute effect. And in the prefrontal cortex, we see that the levels of serotonin spike during the drug epic for the medium and the high dose, and then for dopamine, that happens at the high dose during the drug epic. And then in somatosensory cortex, it's a similar pattern.
Interesting. All right. So you find DMT in the brain. It obviously goes up when you give IV DMT. That's what we just looked at. It actually stays elevated at the higher doses for longer than you might have expected previously, longer than it stays at high levels in the blood. You're seeing here that it's also increasing levels of dopamine and serotonin. And did you guys look at anything else, or just serotonin and dopamine?
We did. We had that whole panel of about 17 different neurotransmitters, and I feel like that might be this next figure here. So yeah, this is looking at whole panel. There's acetylcholine, there's GABA, glutamate, and then a number of different amino acids, histamine, glutamine, glucose. Yeah, just a
Hmm.
number of different things. And
Were there any
the kind of
Yeah,
The big takeaway here is that there were no drug effects. So there were no changes during the drug epoch
I see.
with any of these neurotransmitters or signaling molecules. We did see some changes in the recovery periods. So I think one interesting one is phenylalanine. You'll see decreases in the post-drug and the recovery period in the high dose. And phenylalanine is a precursor for dopamine, so that could relate to increased dopamine production,
Mm.
something there. Another interesting thing is that we don't see any changes in GABA or glutamate, and serotonergic psychedelics, classic psychedelics, are thought to be glutamatergic, and a number of studies have shown that glutamate spikes, especially in the prefrontal cortex, with psychedelic administration, but we didn't see that here. And that actually, to my knowledge, had not been investigated with DMT as of yet. So I know it's been shown in rodent models with psilocybin and other psychedelics, but had not been tested with DMT, and our results didn't support a glutamatergic response.
Mm-hmm. One little question here, from my own understanding. You've got three different doses that you used. Were these completely distinct cohorts of rats, or were you giving the rats the low dose, then giving the same rats the medium dose, then the same rats the high dose?
Yeah, it was a repeated measure, so every rat got all three doses in
Mm-hmm.
a randomized way.
Okay, in a randomized way.
Yeah.
Got
Mm-hmm.
it. Interesting. Okay, so the EEG results. Can you set that up for us? So give people a sense for what is EEG actually measuring, and what do we already know from the literature in terms of the human work showing what's happening in response to DMT?
Yeah, so EEG is I mentioned with the microdialysis measures and the neurochemical analyses, you've got this really poor temporal resolution. You're measuring events that happen over milliseconds in a bin of 12 and a half minutes.
Mm-hmm.
EEG does not have that issue. You're measuring 1,000 times a second. You have a sampling rate of 1,000 samples per second, and what it is actually measuring the kind of summarized or collective electrical activity of neurons across the cortex. It's only measuring cortical activity. So if you look at some studies with fMRI or PET or other things like that, they're actually able to measure subcortical brain activity, but EEG is just focused on the cortex. And different analyses are conducted with EEG. One is kind of the most standard is a spectral power analysis, and that's kind of looking at the electrical activity of the cortex at different frequency bands. So separating it out from the low frequency of delta, about 1 to 4 hertz, up to theta, alpha, beta, and all the way up to gamma
Mm-hmm.
frequencies, which is about greater than 30 hertz.
I see. So if we give people a mental image here, if you imagine looking at the surface of a lake on a windy day, it's very choppy. There's lots of little waves going, and then a boat drives by and it's creating, larger, slower waves. You've got all of these waves happening, little fast ones and big slow ones. And so this is a method for counting how much of each type of wave you have rippling across the brain. Is that kind of a decent summary?
Yeah, that's a really good summary. Yeah, you're kind of measuring the contribution of the little waves and the big waves, and determining how that contributes in terms of overall brain activity.
Mm-hmm. And what's the punchline here? You give DMT. What are some of the more salient changes you see in the EEG signal?
Yeah, so I think the two kind of takeaways was we saw a decrease in theta activity and increase in gamma activity. And it's when you're doing EEG in rats or in any rodent, it's a little bit unique to interpret, a little bit different to interpret in terms of relative to human studies.
Mm-hmm.
So one of the most salient or common features of DMT activity in humans is alpha suppression or suppression of the alpha band, and we didn't quite observe that in the rodents because the alpha band is not as prominently it's not as prominent of a functional band,
Mm-hmm.
in rodents. So it's very important for humans, but not so much for rodents.
Mm-hmm.
But we do see theta suppression and then the gamma activation, and the gamma activation has actually been cited in a few different human studies, so that correlates with some of the previous human research. And we measured gamma activity at a higher frequency than you're generally able to do in human studies. And we specifically saw a high activity in the high gamma range, so above 100 hertz.